All right, first thing is just a little check on where we are with things. So we're in the stretch here. We have several more topics, three big topics to cover to wrap things up here. Today we're going to talk about the life course perspective, and then you all will do presentations on life course next week. After that, we've got living conditions. So we'll be talking about communities, neighborhoods, and housing. So it's more about, where you live and the kind of neighborhood you live in and that sort of thing, and how that affects health. And then lastly, we're going to be looking at things like poverty, racial, ethnic inequalities, socioeconomic status, and how those things are related to health outcomes. So the life course perspective that we talk about today is a little bit different than some of the other two topics that we have. Let me make sure I'm recording. Yes, I am recording. It's a little bit different than the other two topics that we have coming up, and here's why. When we talk about things like your neighborhood and where you live, and things related to race, socioeconomic status, and that sort of thing, a lot of that is going to be more cross-sectional analysis, or a more constrained period of time that you're looking at things. Now, there can be some longitudinal studies that are done in those areas for sure. So, these are not necessarily mutually exclusive, so to speak. But the life course perspective implies that we're looking at exposures and things that can impact or affect your health over some period of time. Whether that's the lifetime, whether that's the first few years of life, which is the example that I'm going to talk about at the end of the lecture today, or what have you. The thing that makes it a little bit different and a little bit unique is this idea of time and the passage of time. And so the life course, for sure, and in particular, lends itself to longitudinal studies or studies over time, where we can follow people over time, usually prospectively. But of course, you could, if you have a secondary data set where people had been followed over time, you can do a prospective analysis, but still, apply the four models that we're going to talk about in particular. So the main points that I want you to take away from this life course lecture today is that there are four primary models in the life course that we want to take a look at. They have somewhat confusing names, and sometimes the examples that we talk about are not exactly clear. So just keep in mind that it takes a little bit of time to digest this information and think about it and read into it, and look at studies, multiple studies, and see multiple examples before it really starts to click. And by the way, let me go back to our course. When you go to our folder for today, which is in the after midterm slides folder under life course, I want to point these things out. So the lecture slides are here. You have the chapter from textbook for this part of the course. The Marmot and Wilkinson textbook has, big picture kind of sociological issues that we're talking about. And so our textbook for this part of the course is not the theory at a glance anymore. It's we've made a shift here. So there's a chapter in there that you can read that some of the examples that I talk about today come from there, and it gives you a bigger perspective. And then I've also got these life course readings. These are short little readings, especially the World Health Organization life course reading. We're talking about the 4 WHO models today of life course. And so that's where that comes from. And I wanted to make sure you had a reading related to that. And you all may remember that I mentioned a week or a couple of weeks ago that one of our PhD students who graduated last year, who's now doing a postdoc, published a paper where she actually applied life course theory to what she was studying. I'm going to highlight that for you at the end of the lecture today, and you have the actual the publication. It was just published this month, that just came out. So that was really, really good timing. So there's some extra readings in here. That's just for you to take a little bit deeper dive, and that's supplementary stuff. Most of what you need to know, of course, is going to be coming from my lecture. All right. So health is the product of risk behaviors we engage in, the healthy behaviors that we engage in, and the environment that we live in, and how these things interact with each other through different periods or phases of our life, and certainly across our lifespan. And so some of these exposures that we have, some of these behaviors that we engage in or not, these factors can have a one-time effect on us that maybe lasts for some period of time or even a lifetime. Sometimes these things interact with one another, where one exposure or one thing that happens to you early in life and then something else that happens later can compound one another. There's different ideas at play here that we'll be talking about when we go through the life course models. And so when you're doing any kind of longitudinal study, and it really doesn't matter if you're looking at biological variables, if you're looking at psychosocial variables, interactions between people, psychological variables, et cetera, and so on, you can apply these life course models to studying these factors. And sometimes you have a mix of these things as well. Now, what we've got here in this figure is sort of the idea of health and vitality, if you will. And we've got 2 different people that are represented here, one by the blue line and one by the orange line. So the real takeaway from this is that as a person increases in age and their growth and development, their health changes over time. Sometimes it becomes more positive. Sometimes there are negative outcomes that happen along the way. But we all have these sort of curves that represent our health status over time. And you can see that one of these individuals has a much steeper decline in their health over time. That would be the orange line, than the person with the blue line. Another thing that's important to highlight and talk about with this image is that we've got this so-called disability threshold that's represented here. And notice that even though both of these individuals' health will decline over time, and that happens with age, and that's normal, one is more significant than the other, one being a decline so great that they have some disability in the later years of their life where the other person does not. So we all know this and understand this, and we can think of real life examples of this with grandparents or people that we know from the community or what have you. But this is a really good place to start when we're talking about the life course model, because we're going to see a lot of graphs and figures that are represented in this way. So the life course perspective emphasizes this idea of time, as well as the social environment that we live in and the things that we're exposed to in our social environments. So when you look back across a person's life, whether that be an individual or a group of people, a lot of these longitudinal studies are done with a certain kind of person to look for long-term implications of lifestyle and health outcomes, and these sorts of things. So, as an example, some of the early longitudinal epidemiological studies were done with doctors and nurses. So you may have heard of the Harvard Nurses Study, for example, that was done many decades ago. And that's kind of a captive audience. It's a group of individuals, a cohort, if you will, that share a commonality in terms of their education and their occupation and that sort of thing. So in some ways they do have similar exposures, at least from an occupational standpoint. There are studies that have been done. I knew a guy who was doing a postdoc when I was at University of South Carolina, who was an epidemiologist, and he went out to Utah to study Mormons. So Mormons, if you don't know, they are really, really strict about not drinking, not smoking. They have a generally healthy lifestyle. And so studying that group that happens to be a group with um, um, spiritual belief ties or bonds, if you will, they do have these commonalities in values that they hold with respect to abstaining from drinking, abstaining from smoking. They generally eat healthy and are physically active and those sorts of things. And so studying a cohort like that is almost the exact opposite of the typical American cohort in some ways. And so it's kind of interesting to think about studying a group that has a lot of positive things related to lifestyle factors as opposed to what we typically do. But in applying this life course approach and these models we'll talk about today, we are interested in any number of hazards, or the epidemiological term, of course, is exposures from the physical and social environment. And so that could be things that happen before you're born. It could be things that happen in early childhood, in adolescence, during adulthood, and of course later in life. And so we want to know how different exposures from the physical and social environments during these different time periods and across these different time periods will affect, chronic disease risk, our whether or not we die early, whether or not we have some sort of morbidity in terms of inability to engage in activities of daily living and those sorts of things. And the idea here is to identify relationships between these exposures and these time points that are meaningful, and to understand how something that happens to you at one point in time affects your health, how multiple things that are not related to one another but are still bad for your health, if you will, relate to health outcomes, how things that are related to one another that maybe happen close together in time or spread out in time, how those things affect your health. Is it additive? Is it multiplicative, etc.? Those are the kinds of, there's so many different kinds of interesting questions that you can ask when you put all this stuff together. And so the life course models provide a good structure for you to operate from in terms of deciding how to look at the relationship between various factors. So this is kind of a little aside here, but an important point that I want to make. So this is not really theory, as we've talked about it earlier in this class. This is really more of a systematic and organized way of thinking about how things fit together and work together. So it gives us a structure to work from. It doesn't necessarily predefine how your variables relate to one another, but it gives you a structure, a logical structure, where you can flesh out the relationships, whether the insults or the hazards or the exposures, whatever the language is that's used in the particular paper that you're reading, really all mean the same thing. How they relate to, how they can potentially relate to one another, logically speaking, are they independent, are they correlated, etc. This gives us some structure to work with. And so in some sense, I like to think about the life course models as a way to organize all of these factors that we may know about from our clinical knowledge, from the published literature, etc. And it gives us a way to put them together in a meaningful way that we can test different types of relationships, knowing that there are some potential ways that they can relate to one another, but yet we're not defining those in any sort of way. So we still got the flexibility to sort of move the chess pieces around on the board, so to speak, and see what happens. But we do have some structure or strategy to begin with. All right, so again, I've already mentioned that longitudinal studies are very useful if you're interested in studying life course and using those models to structure the things that you want to look at. And there are a number of well-known longitudinal studies, and just a few that are done with children. As examples would include the longitudinal study of Australian children, the Millennium cohort study in England, and then here in North America, we've got the National Longitudinal Survey of Children and Youth. That's in Canada. We also have one called the HLTH study that's done in the United States. That's one that I don't have listed here. But I highlight these because they're focused on children, and they start at a very early age. And depending on the particular cohort study that you're looking at, they may follow people all the way till death. They may follow them into early adulthood. It just depends on how long these things continue to be funded. And so basically the way these things start is that you'll have a group who gets funded to do an initial longitudinal study. They have some success in what they do. And then over the years, literally over the decades, they get additional funding to continue that work and extend that work. And that usually leads to a lot of people maybe at one institution where that particular study is housed. Like the University of North Carolina, Chapel Hill is really the home base for the HLTH study, which I mentioned but is not listed here. And so essentially what you have is, once you have that kind of thing in place, you have people from different disciplines that can plug into that and be part of that. And then over the years and over the decades, you have new people in terms of doctoral students, postdoctoral fellows, new faculty coming and going over time, and they can get involved in that. And these studies just go on and on and on, and they usually track individuals or families over time. As you would imagine, these kind of cohort studies, longitudinal cohort studies, require a lot of coordination, a lot of work, a lot of planning. Takes a lot of resources to be able to get one of these up and running, and to keep it going. See, we've got someone has a question here. So Jennifer just posted a link to a study that looks at happiness over time. So that's a good example, Jennifer, of there would be multiple papers that come out of that particular project, and you could highlight one of those papers next week. Oh, we've got some positive chatter going on with that one. That's an example of one that you might highlight next week, by the way. Any paper that you can find from the longitudinal study of Australian children, Millennium Cohort Study, or the National Longitudinal Survey of Children and Youth in Canada, or the Ad Health study that I mentioned, those would be other examples as well. So it's really hard to, these kinds of studies are really big and complicated, and they come with a considerable price tag. We did have an epidemiologist here a few years back who has now moved on to UCLA, and several of us wrote an NIH grant with her. And you know, the big NIH grants that you get as an individual investigator with a team, of course, are in the ballpark of a couple $1 million. And those usually occur over a 3 to 5 year period. This longitudinal study had a price tag around $10 million. And it was going to go for 7 or 8 years. I don't remember. Can somebody close that door, please, to the kitchen? Okay. Okay, but there's noise from the kitchen too. These things are super, super expensive, and they just go on and on and on. But we get lots and lots of really good information from these. All right, so moving right in here. Let me double check the chat one more time, make sure we're good. Okay. Here are the 4 models that we're looking at today. The 4 models that are outlined by the World Health Organization. You have a short reading on this that you can look at, but I'm going to hit the high points for you today. So the first model is what's called the critical period model. And that one is very simple and straightforward. It's the basic model. It's where we start. And what that means is that there's some time period, let's say early in life, where something happens and that affects your health, or your physical ability, or your mental ability, or what have you, over the course of your life. Next, we have a critical period model with later effect modifiers. Now that's starting to sound a little bit fancier now. We're using terms like effect modifiers, but the meaning is implied in the language here, right? So an effect modifier, what does that mean? It means that something happens, and then at some point in time later, another thing happens which exacerbates that first thing that happened. And so those two are sort of building on one another, if you will. Next, we have two more. So we've got 2 critical period models, and then 3 and 4 are accumulation of risk models. So this means that things are happening at multiple time points over time, usually. And so the first accumulation of risk model is with so-called independent and uncorrelated insults. So that means that there are a number of things that happen to you over time. Those things are not related to one another, but together there is this so-called accumulation of risk. Sorry, my dog is demanding that I pet her right now. She always does this when I lecture. She wants me to be quiet. She's tired of listening to this nerdy stuff. That's what it is. Okay. Go play with mommy. And then we've got the accumulation of risk with correlated insults. So these are things that are related to one another, or build or spin off of one another. And that one's sort of like can be a really nasty model, because you have almost this exponential effect that's going on, because there are multiple things that are happening to the person, and a lot of well, not a lot of times, but all the time, these things are related to each other in some way where one leads to another. And it's almost, you know, I kind of envision like a snowball that's going down a hill that's just getting bigger and bigger and bigger. So that's my simple way of thinking about it. So let's kick off with the critical period model, and I'll have a few slides on each one of these with some examples. Most of these examples, by the way, you will find them in the chapter reading from the Marmot and Wilkinson book. Some of these I've pulled in from other places as well. It's kind of a mix, but so the critical period model is sort of based on this idea of, you know, if anything goes wrong, permanent damage can result. So we think about maybe one singular event that happens and it just kind of modifies your health trajectory in some way from that point forward. So think about something sort of dramatic here, like a person who is in a car wreck and is injured and is physically damaged in some way where they don't have all the function that they can't reach their potential that they could have had, and they can't do all the things that they did before. So maybe some kind of damage to their legs or their arms or something like that. Think about a person maybe falling and hitting their head from whatever reason, riding a horse, riding a bike, I don't know, falling off a ladder. And there's some mental disability that occurs related to that. That would be another really good example of a critical period model. So it's not necessarily true, but sort of the easiest way to think about something like this is, other than that one thing that was pretty dramatic and bad that happened to the person, you know, they're pretty healthy, otherwise speaking. So there's this one thing with the critical period model that just really stands out as this is the thing that has caused this disability or this problem or this health issue that a person has at that point forward, moving forward in their life. So it's an exposure or insult. There's multiple different terms that are used. The term hazard, you'll see that as well. These things have a lasting effect on that person and on their health or their physical or mental abilities. So one of the things that one of the things that I wanted to point out about this particular model, this kind of base model, if you will, where we sort of start from, is that you can look at issues like this even in a long-term study. And if you can identify certain exposures or hazards or insults that occur to someone, you're really going to kind of hone in on this one particular period of time. And a lot of times what happens immediately after that changes their trajectory, just so you can get a better understanding of the impacts of that. And then there's other related questions that you can have in terms of how those things might affect your relationships with other people, your ability to drive, you know, anything that we can think of moving forward. But this is the most cross-sectional thing, which is sort of a, you know, preset point in time, if you will, or I should say constrained point in time, where we're looking at things with a little bit tighter lens than we do with some of the other models. So in this British children's study that is mentioned here, they're looking at basically allergic symptoms and asthma symptoms. So these atopic symptoms can be, you know, skin rashes and things like that, but also things like asthma and wheeze that are typical in people with asthma. And basically what they were able to do is to look at combinations of exposures or insults or hazards, if you will, and how those combinations can classify or categorize how healthy a child is at different points. So we have one category here where we have children who have low wheeze, so they don't have a lot of wheezing, and low atopic symptoms. So this is we would think of this as the healthiest category. Now, keep in mind that all of these we're looking at the living environment. So that's really the exposure that's of interest here. And then these categories of 2x2 table, if you will, of symptoms, combinations of symptoms are really the outcomes that are being observed. So we also have another category of low wheeze, high allergic symptoms, high wheeze, high allergic symptoms, high wheeze, low allergic symptoms. And so you might
Did you name drop the book?
Who's got their mic on?
Mm-hmm.
So you might think that the high wheeze, high allergic symptom group, which does sound to be the worst, you might think that that would be, the poorest, the most disadvantaged, and that sort of thing. It doesn't turn out to be that way. What matters most is the dampness that you're exposed to in your home environment. That's going to be more strongly predictive of being high on both wheeze and allergic symptoms. So it really doesn't matter so much how much money you have. It's just this dampness is very important. The most advantaged and damp homes during infancy. So these are people with resources or money, but they have damp homes for whatever reason. We can see a clustering of low wheeze and high allergic symptoms in that group. And then what we would think of as the most disadvantaged group, the lowest socioeconomic status, when you drill into this particular paper, you'll see that they have a lot of asthma, but the skin allergies and those sorts of things are not as bad. And so maybe as a researcher, you go in with some preconceived notions as to how these combinations will look in terms of exposure, and you either prove that or you disprove that, or you don't know, but you want to be able to classify or describe health outcomes related to combinations of these things. This would be an example of a critical period model where we're looking at the housing environment where you live when you're young. So that's a really good example of a critical period model. So we can think about critical period models again as happening at different time points throughout a person's life. So with kids, of course, we're going to think about the young ages. Maybe immediately after, right after they're born, you're going to see a lot of studies that are looking at newborn health indicators, certainly during childhood, during adolescence, but also in early adulthood and middle to late adulthood, middle aged. And then in older ages, we can think about all those different time points as more constrained critical periods. So I wanted to make the point that we're not necessarily always looking at kids and the early life. We can look at different time points. So remember, we've got 4 models. And I want to go through those in turn here. So we have a critical period model. The first one here is with or without effects of later life risk factors. So the exposure that we have here is low maternal folic acid status around the time of conception. That's a nutritional dietary intake issue. If mom doesn't have the appropriate diet or she's not taking prenatal vitamins. And that exposure to the developing fetus could be an insult that could then follow into neural tube defects after they're born. So that's a good example also of this critical period, the basic critical period model. Next, we've got critical period model with later effect modifiers. So what that means is something happens early on, and then later in life, another thing happens that makes their health outcome worse. So this suggests that a person who experiences some hazard or negative exposure during a specified period of time that has a lasting effect, and then things that happen later in life make that worse. That's just restating what I just said. So one study was done looking at birth size. So how big are you when you're born, and type 2 diabetes risk. And this was a Swedish study that was done back in the mid 90s. And it was a longitudinal study of men that were born in the early 1920s. So they have information, medical records on these men and their birth weight, their birth size, other clinical indicators, but they're studying them later in life, which typically after 40 is when you develop type 2 diabetes. And so they're following them later in life. You can see here that they did glucose tolerance tests with them at ages 50, 60, and 70. So a person with a low birth weight, they have higher risk for coronary heart disease, high blood pressure, and insulin resistance. But this can be altered if you maintain a healthy weight during your lifetime. So we don't, we may not often think about this issue of low birth weight being associated with type 2 diabetes or coronary heart disease, because you're talking about a skinny baby will be healthier when it comes to things like chronic disease, right? Well, that's not necessarily the case, and that's well documented. And so that's the working hypothesis that they have here. So they looked at the blood sugar levels of these men when they did a glucose challenge test at the ages of 50, 60, and 70. What they found was that diabetes was more prevalent in men who had a low birth weight, and diabetes was less prevalent in men with a lower BMI as adults. So this suggests that if you are low birth weight, you already have the deck stacked against you in terms of your risk for diabetes in this case. But if you have a high BMI later in life, it's going to make that worse. If you have a low BMI later in life, it helps to counterbalance that or counteract that. And so they're looking at more than one event that happens from in this case, at birth and then later in life. Now, I just kind of skimmed the details of these things just to make the point of how this matches up with the models. I don't get into a lot of details of these, but if you need any references to any of these, if you don't have sufficient information there, I can get you more. So this is kind of a visual representation of what we're talking about with the critical period and later effect modifier model. This is looking at basically something happens that puts you at risk for arthritis, in this case, rheumatoid arthritis. And what they're looking at, and so that would be the first exposure, if you will. What they're looking at is how long you wait before you're diagnosed and receive treatment. The diagnosis, just kind of forget about that for just a second. The key thing here is, when do you start treatment? And so the later effect modifier then is how soon or how late you start these. The DMARD is disease modifying anti-rheumatic drugs. I remembered it. I can't believe that. That's what that stands for. So those are drugs that are given to people with uh, rheumatoid arthritis, for example. And what it does is it slows and prevents and can even, in some cases, reverse that disease. So the timing of that treatment is very important. As you would imagine, the earlier you start that treatment, the better your health outcomes are going to be. And so when you look at over time, we're following this from left to right over time. You're looking at the progression of the disease. And so what happens is if you have early treatment, your progression is going to be far less. Your arthritis is going to be far less than someone who did not receive treatment, and also much better than someone who received treatment late, so to speak. Let's see, I think we've got a question here. Okay. So the effect modifier here then is when you start or whether you start treatment. So we've got early start, late start, and then no treatment at all. And this is just an image for us, another way that we can think about that. All right, so now we have critical period with later effect modifiers. Now we're looking at things that happen again. We start out with low birth weight, and then that results in a poor motor system. Because of that poor motor system or ability to ambulate and get around, that predisposes us then to a less active lifestyle. And of course, being less active, if we decrease our activity, even if it's from something like a disability, now we have this compounded issue of something that happened to us. It prevents us from being sufficiently active. And because our activity levels are low, now we're at greater risk for chronic disease in the form of cardiovascular disease in this example. So this is where something that happens early in life that we can see a clear impact from leads to us not having sufficiently healthy lifestyle that we engage in. And then, of course, that leads to this risk for cardiovascular disease. So accumulation of risk models. Remember that this is number 3 and 4, so there's 2 of these. The accumulation of risk model takes into account this idea of adverse exposures that a person faces that accumulates over a lifetime. And so these 2 models in particular really work well, these 2 more so than the other 2, at looking at a longer period of time for an individual or a group of individuals, because there's this idea of accumulation. So we have with independent and uncorrelated insults, and basically that means that there's bad things that happen to you over time, but they're not really related to one another. But each one of those things can contribute in some way independently to your health status, of course. And then we have the correlated insults model, and that's where things kind of cluster together in such a way where maybe one exposure or lifestyle factor kind of leads to another, and they really kind of build on one another. And that's this idea of that that snowball that I was painting earlier. In this first study by Ching et al., they were looking at cigarette smoking, alcohol consumption, and the risk of nasopharyngeal, so upper respiratory cancers, if you will, in people that were living in Taiwan. And basically what they looked at in that study, and a lot of times we think of cigarette smoking and alcohol drinking going hand in hand, but that's not necessarily true, and that's not really what they were looking at here. But they basically were looking at people who smoked and how, and that sort of direct risk that comes along with smoking and these types of cancers. They also looked at people who were exposed to secondhand smoke and how that relates to that. People who drank and didn't drink. This one's not as clear of an example, but this is one where each one of these things could by themselves um, be the primary thing that's causing the cancer, but they're not necessarily related to one another. With the correlated insults model, when you look at that Japanese study, one of the things that they looked at is the outcome of interest that they were interested in there was risk-taking behaviors. And what they found, I'm just going to really kind of focus on this bottom bullet point, is that women who were lower SES were much more likely to divorced or be divorced, to be unemployed, and a number of other things. And it's almost like a domino effect. It's where these things sort of clustered together. All of these negative predictors kind of clustered together. These series of characteristics or life events or what have you. And the more of these that an individual woman had, and the more intense these things were, the more likely she was to engage in some type of risk behavior. And I don't remember specifically what those risk behaviors were, but this is one where you have these different exposures that are indeed related to one another. Sometimes one kind of initiates another to happen, if you will. And so those things are very much related to one another, and they all together in some way contribute um, cumulatively to this idea of engaging in risk behavior, whether that's frequency of behavior or just engaging in one really big event, if you will. So this is what accumulation of risk looks like. This is how we can think about it. So you might think of the bottom line here as maybe one potential trajectory that a person would have, and on the y-axis, of course, we've got the development of non-communicable disease. So that's the outcome of interest here. And then the top line that we have there would be another either real or hypothesized trajectory that another person could have. And then we see a series of things that can happen here: exposures, insults, um, hazards that could be related to causing that accumulated risk. And so what we're seeing clearly from this picture is the difference between one person's trajectory, or the average trajectory for one group. You can think of it that way with the bottom line, versus the a worse trajectory with increased risk for non-communicable diseases on that top line. And the difference between those two, that difference in between, when we talk about accumulated risk, that's what we're talking about. So it's the risk that happens to that person on that top line above and beyond some baseline level, or like the normal person's life, if you will, however you want to say that. But it's the additional risk that is accumulated by that person, or on average by that particular group of people. And this is what it looks like. Accumulation of risk models. Continuing here, we've got independent and uncorrelated results. So the outcome of interest here is early menarche, faster growth rate, presence of stepfather, war. That seems totally just kind of a weird thing to throw in there. These are three things that are known to be related to early menarche. You may not have thought that war might be related to that, but it is. And those are the weird kind of things that people in Europe study. That's where this came from. But these things are not related to each other in any way. So if you have a stepfather present, that doesn't have anything to do with a faster growth rate that you have, biologically speaking. And these things are just not related to one another. But if these things happen, um, to the same individual, each one of these increase the accumulation of risk for a particular individual. So this is a good example because, as odd as war sounds in terms of a factor that we might be looking at, this is clearly things that are not related to one another in any sort of way. And the idea here is that there are things that are observed that we know that if these things happen, it can be related to some health outcome or condition. In this case, we're talking about early menarche, but these are clearly not related to one another. I think there's a question in the chat. Let's see here. Is it connected to the social ecological model of looking beyond the person's characteristics for a health behavior? Yeah, so these would be, well, the faster growth rate, I mean, that's physiological. It's, that's your genetics, essentially. There's other things that can play into that. There's other variables that could be at play there. But then the presence of stepfather, this would fall into the social environment realm, and then war could be, there could be stresses that occur as a result of that, from the damage to the physical environment. If you think about pictures that you've seen of like World War II, where people literally had to walk through the streets where there were crumbled buildings and that sort of thing, to the social disarray that occurs because of war. So yeah, in some ways you could think about how the growth rate is really physiological. It's within the person. If you have a stepfather, that's something that's in your intrapersonal realm, if you will. And then war is just going to be, outside the person, that maybe more than one level. But this is a cluster of things that have nothing to do with one another. And that's the main point to make there. All right, accumulation of risk models continued here. We have correlated insults. So these are things that are related to one another. And then we have different flavors or categories, subcategories of that, if you will. So this is where things get a little bit crazy. But we have risk clustering models, we have additive models, and then we have trigger models. So risk clustering models, if you are a child within a low socioeconomic status family, things like poor growth, parental divorce, and poor diet are fairly typical, generally speaking, for people who grow up that way. And it has been shown that those things together can be related to earlier menopause later in life. So we're looking at things that cluster in childhood, but things that are seen, basically in midlife, if you will. And that's the risk clustering model. That is one example of accumulation of risk model. The next example that we have, or subcategory or submodel here, is this so-called additive model. So smoking and early menopause, those things have direct relationships to your risk for cardiovascular disease, but they also, smoking can increase the chances that you experience early menopause. And so there's a pathway there as well. So these things are more related to one another. At least one has an impact on the other. And then the chains of risk submodel that we have here: risky sexual behavior in adolescence that leads to sexually transmitted infections. And then the outcome of interest here is infertility. This would be, you should recognize this as what we have referred to as a mediating model, right? So there's something in the middle that happens where one factor affects another, and then the relationship between those two have a causal relationship that flows forward into some outcome that you're observing. So don't let this get you too, let your mind spin too much. But just know the general difference between these four models. This is going a little bit further just to show you that you can expand these things even more. So a little bit about social structure in the life course. So we have to remember that we have biological and physiological processes. We have genetics that are at play, but we live in a physical world. We live in a social world, and all of those things can interact with one another. And that's where things actually can get really, really interesting in our field and the kinds of stuff that we study. So we've got some examples of cross-sectional studies here and longitudinal studies here. So there is this idea that the context of how you grow up or how you live has an impact with with respect to having advantages or disadvantages. So we can think about health disparities and people, the haves and the have-nots, those sorts of things. People that have income and resources and education generally have better outcomes than people with less education that live in poor neighborhoods that don't have as much education and opportunity. So those contexts that we live in, those circumstances of our life, we can look at those things through a cross-sectional lens or a longitudinal lens. And so just a couple of examples here for you. A cross-sectional example would be a person whose working environment is free of hazards, is likely to reside in good quality housing, to live in an area with little air pollution, and to have an income that permits a varied diet. Those things, as a general rule, fit together. Someone exposed to significant hazards at work, so this would be sort of the opposite of that, is likely to have inadequate housing, to be exposed to more pollution, and to have limited income and less dietary variability as a result. So we can look at those things at a more specified or constrained point in time. Longitudinally speaking, a couple of examples. The first one, a child from an affluent home is more likely to succeed academically. They'll have a better job. They'll have good income for retirement and security later in life. A child from a disadvantaged home is more likely to struggle academically, have fewer job options, and as a result, have less income and security and savings for later in life. Now, that's not true in every single case, in every single scenario, but we do know these things operate together in a way that is predictable. So knowing those sorts of things, we can use that information as a way to help structure how we're looking at variables. So whether or not you're creating your own model as you did previously, or if you're applying one of these life course models, you can use that information to come up with better research questions, richer research questions, and so on. So just to kind of begin to wrap up this piece here before I get a little bit deeper into that example that I wanted to share with you, just look at this health trajectory where we've got the light blue line and then the dark blue line. And really all that I'll say about this is that we have two people who start out not necessarily in the same point, but depending on what point in life you're looking at, one person is doing better than the other person with respect to their health status. And they cross over a couple of times here. So different people have these different life circumstances and things that happen to them along the way that change their trajectory. And so our trajectory is not necessarily this smooth curving kind of thing. It can go up and down. It can have a generally increasing trend and then decrease. everybody has their own trajectory that would represent their health over their life course. But depending on the things that happen to you and how many things happen to you and how positive or negative those things are, your trajectory, your individual trajectory is going to change. We can also use this concept that we have and extend it beyond comparing two individuals over their lifetime, and think about comparing people that live in the United States versus people that live in, I don't know, Sri Lanka, just some other place in the world. We could compare men and women. We could compare high socioeconomic status with low socioeconomic status. We can look at high birth weight versus low birth weight people. Going back to one of our previous examples. So we can think of these lines as representing individuals or groups or cohorts, if you will. And of course, the group or cohort trend line is going to be one that's sort of the average of what happens to the people in that group, as opposed to looking at individual trend lines, which are going to be far more precise because we're talking about one person at a time. So a few critical issues to mention before we wrap up here. Critical periods: when or if anything goes wrong, permanent damage can result. That's a quote that came from your reading, and I've included it here a couple of times. It's not just when you're developing, but also early on, right after you're born, childhood, adolescence, all the way through life. There's any number of things that can happen, and there's multiple critical periods, as I've already mentioned, that we could look at, and we can constrain our research questions and evaluation to. Critical social transition. So, a person who starts out poor may not end up that way. They may, some of the richest people that you can think of maybe didn't start out that way. They started out with the deck stacked against them, if you will. And so you can think about how different transitions happen to individuals over time. Really kind of looking back at this figure here, we've got positive and negative things that are happening to these two individuals if we think of this as representing two different individuals, and how there are certain transition points and time periods along their life where their health status is improved or sort of regresses, if you will. So there's a number of issues and transitions that can be thought of and implemented into whatever it is that you're studying using the life course models. All right, so life course application here. You do have this PDF file in your reading. Ellen Francis, again, was one of our PhD graduates. She came to us with a psychology background, and she basically earned an unofficial human nutrition / biochemistry degree because of the kinds of things that she was studying. And so that was the expertise or content information that expertise that she picked up along the way. But in terms of methodology, she's definitely uses a lot of epidemiological methods in her research. She's now a postdoctoral fellow at the University of Colorado, Denver, at their medical school. And the center that she works in, it's called LEAD, LEAD. And I can't remember. I think it's life course epidemiology and diabetes. I don't, those words are in there somewhere, but they focus on diabetes and they actually focus on the life course. So it's not surprising that she was interested in applying the life course models to what she was studying. And one of the things that she looked at in her dissertation was, maternal um, nutritional status and how that relates to a variety of uh, outcomes of the children that are born to those women. And in this particular study, I just pulled some things from her paper. Um, they wanted to look at mom's blood sugar level during pregnancy and how that relates to their baby's fatness and metabolic health. And so they were specifically interested in the child's blood sugar levels, insulin sensitivity, and those things that would be potentially related to diabetes risk later in life. And they wanted to, and they tracked kids, by the way, all the way through 4 to 7 years of age. And so this is something that happens with longitudinal studies. You have kids that come through in a batch over a several year time period. And so, you have these bounded ages of 4 to 7, and that's where that comes from. But they're following the same kids over time. So they wanted to know if a mother's hemoglobin A1C, which is a stable, way of looking at blood sugar levels over really a couple of months period of time, as opposed to just what's my blood sugar level right now. And so it's a really good indicator of where a person's blood sugar levels have been over the most recent past couple of months. And so they wanted to know if that was related to offspring, to the children's size, to their fatness, and also their own blood sugar levels. And they wanted to see if a child's fatness or adiposity mediated that. And that's really kind of the long and short of what they were doing. So they have this cohort of 1,400 that they're working with, and you can see how we get this dwindled down into the final sample size. So women with offspring whose child attended a research visit, so about half of those that they started with. Women with hemoglobin A1C measures at 27, around about 27 gestational weeks. They retained most of those, so they lost about 77 there. And then they've got neonates from those women who had measured fat, their percent fat mass was measured. That gets us down to 614 because we've got the 87 over here that are excluded because they didn't complete body composition measures. We have more from there that were excluded because they had low percent fat mass, and then mother-child pairs that were missing data. 263 of those were excluded. So they end up with this sample of 345. So this is not unusual to start with, a large sample and then you dwindle down to whatever you get. But this tells the story of what happened in terms of this particular study. So let's look at some of the things that were measured and think about factors for a minute. So the exposure of interest was maternal blood sugar levels during pregnancy. And again, that was measured with hemoglobin A1C levels. The outcomes that they were interested in were various biomarkers of the offspring, and particularly ones that look at glucose and insulin variability. So fasting glucose, insulin, and insulin sensitivity were three things specifically that they measured with the children. The mediators that they looked at were measures of child adiposity, so body composition, fat mass, and fat-free mass. And then the covariates that they included in their model were maternal pre-pregnancy BMI. So mom's height for weight was controlled for. That's what covariate means. So here's what they're looking at. They have these two models that are being tested here. So this is a direct effect model that we have up here where we're looking at the exposure of mothers' blood sugar levels at 27 gestational weeks, and then these biomarkers within the children that they were looking at. So these measures of insulin, of pancreatic function. That's what the HOMA 2B looks at. Um, Insulin sensitivity, fasting blood glucose. Down bottom, we have this mediational model where basically we've got the same thing that we have above there, but we have this addition of these potential offspring mediators in the form of various measures of body fat or adiposity. So they want to know when they add that to the model. And by the way that you refer to this pathway A here is this is the exposure mediator pathway. And then when you look at pathway B, that's the mediator outcome pathway. And so they also can still look at the direct effect between mother's hemoglobin A1C level and these measures of glucose tolerance and insulin sensitivity in the child, even though they look at the mediator. But what's going on here with this mediator is you want to know if child adiposity is accounting for, and if so, how much of the relationship between mother's blood sugar level and the child's these biomarkers with the child. And so if there is something going on, if there is some mediation going on, that direct relationship will be diminished in that bottom model compared to the top. And then you can actually compare the two, this C prime pathway to the C pathway here, to look to see if there's any clinically meaningful or statistically meaningful difference there. If there's mediation, there will be a difference there. If not, then there's no real difference there. So what they found was that children who were born to women in the highest tertile of blood sugar levels during early to mid pregnancy had higher fasting glucose, children with higher fasting glucose at age 4 to 7 years old. So there is some relationship there between mom's blood sugar ability to control blood sugar and these biomarkers of glucose and insulin sensitivity in the children that they were studying. So there was a trend there, but it was not statistically significant. There was no observed mediation in terms of the child fatness in that relationship. And so, you might think, well, that's a bummer. This is just a failed example of mediation and of this potential pathway that we were looking at here, but that's not true. What that means is they've been able to prove, at least in their sample, that there's no mediation here. The adiposity doesn't add that much more to just knowing what mom's blood sugar levels are during pregnancy. That's way more important. It's also easier to track and monitor because it's just done automatically as part of routine clinical procedures. And so that's, from a practical perspective, that's actually good news. But what it also says is that there are other things that are going on here aside from child percent fat mass that are at play. And so now we need to figure out what those things are. So this is not, at the end of the day, you don't say, well, this is a bummer. We didn't find what we thought might be going on. That's not necessarily true. What it does suggest that there's other things going on. Also, one other thing to keep in mind is that this was a fairly small study with, was it 347? 45, sorry. So, doing research with a larger sample would definitely be beneficial. Um, and doing multiple studies to see if you can confirm these same findings, uh, or if you see variability in outcomes from one study to another. So it really takes the accumulation of research to tell the full story over time. But this is a really good example um, of the use of looking at critical period in particular as a model for really a more clinical epidemiology type of study. So these kinds of models are not just limited to looking at social factors or psychosocial factors. This one is clearly clinically based, but we could also, if you had a secondary data set, like some of the ones that we talked about that maybe included clinical data and social variables, we could look at some unique combinations there. Let's see, make sure that there's no more questions here in the chat. Could any of the four models, I didn't answer that one, from who we talked about today be studied both quantitatively and qualitatively? Yes. Most of these are typically quantitative studies, Olivia. But that's not to say that you might want to collect qualitative data around some of your key variables to help explain the quantitative relationships that you're finding in your study. So without that qualitative information, you wouldn't always be able to say, why do we see or not see a relationship here based on our quantitative variables that we have. And so you could definitely incorporate qualitative methods to help paint a fuller picture of what's going on. You could also use qualitative data collection to help you sort out what needs to be studied. And then you apply one of these models in a more quantitative way. Um, but typically these are typically quantitative studies as a general rule. But let's say that so we can talk about the order in which you do mixed methods. Mixed methods means that you're using qualitative and quantitative methods in a single study. So I've described one where it's simultaneous, where you collect qualitative data to help explain what it is that you're finding quantitatively, maybe at the same time. You may use qualitative first to help you decide how to do your quantitative analysis. But then you could also do a quantitative analysis with a follow-up to understand on the backside, why are these people clustering in these different categories with wheeze and allergy symptoms, for instance. Let's get a better understanding of what's going on there. Going back to one of those examples, where we're using qualitative methods after our primary analysis being quantitative oriented to help us understand the findings. And we have a class on mixed methods, by the way. That is the Research 2 class. And it's really good. All right. Well, that wraps up today. What do we got? 6:54. That's not too bad. Okay, so I have been super busy, and I am doing my best to try to wrap up grading. I know that people are going to have questions about that, so just bear with me. Trying to get through this week. I have several extra things that have eaten up my time over the past 2 weeks, and several deadlines I'm trying to meet. So I'm working towards that. So just hang with me. All right. If anyone, this might be next week, when you do your case study presentations, this might be one of the topics where you go, I'm not really sure if this is life course or not. If you want to bounce if you want to run your article by me, let me take a look at it and see if I can give you some input, a thumbs up, thumbs down. The biggest tip I can give you is do an actual search for a life course in the title of the article. That's the easiest way. And then, maybe try to combine life course with, if you're interested in pediatrics or kids, make sure you throw that search term in there. If you're interested in a certain type of disease or disability or condition, throw that in there. And then look at the combination of those on PubMed as I've shown you in the past. That's a good way to bring all that together. All right, does anybody have any questions or comments before we sign off today? I am sitting here in the dark, aren't I? Okay, thank you everybody for hanging with me. Just a couple more topics. We're definitely at the moving close to the bottom of the hill here. All right, everybody, be healthy, be safe. No coronavirus. Take care. See you next week.
Have a good one.
Bye, Sarah. Good night, Sarah.
Bye. Thank you.
Bye.